Well, it's a pleasure to be able to contribute to Genetic Virtual Week and to be a keynote speaker for this session. I want to thank the folks at LabRoots, Lynn Brainerd in particular, for all the help, for Ty and behind the scenes recording this, and all the elements to make this such a smooth process. I want to talk a little bit about precision medicine. I chose the title Moving Precision Medicine from Exotic to Normal because I think we need to stop thinking about precision medicine as something exotic and think of it as something normal. When we see a giraffe, we're quite excited and it's out of the ordinary. When we see a horse, it's interesting and we love seeing them, but it's not quite as out of the ordinary. And that's where precision medicine is going. We're seeing a lot, both on the genomic side and with other tools, becoming normal, just becoming something that's part of the way we manage patients, part of the way we identify risk, that sort of thing. So I'll talk on that piece a little bit. I want to, first of all, have some disclosures. I'm on the board of directors of several companies that are listed there. Scientific advisor to one company. Speaker, done some talks in the past, although it's been a few years, for Genentech and Illumina. And then I'm in debt to my wife. So the problem as we know it with precision medicine, it wasn't that long ago where the main focus was around biobanking. And we talked about precision medicine, but really we were talking about taking tissues, taking blood, taking other samples, tucking them away in the freezer. Then we started linking them with a data warehouse, so the electronic medical record or some other set of data where we had those samples. And then we started getting into some practical applications, some pharmacogenomic applications, but only the minimum. Some tumor sequencing with just a few genes because you had to expand out to more genes with sequencing, start doing prenatal, non-invasive prenatal testing that started being highly applied across many areas of the world in terms of genetics and genomics. And then things have exploded where you see lots of examples, transplant genomics, pharmacogenomics for a number of indications that we'll get into. The neonatal intensive care units now, many of the high-level NICUs have a 24-hour turnaround whole genome that is being applied to identify the risks to these kids and intervene right away. Molecular microbiology is a major advance, and we see more of that coming along. And so precision medicine has gone from an idea, something we may do, something that's more research-oriented, to something that is now being applied on a routine basis in many elements of our life. And if it's not in your area yet, it will be soon, because we're seeing a rapid distribution of being able to apply precision medicine in most parts of the world. Now, taking a medication-oriented approach, the clinical problem is really a wonderful problem. And that is that for most diseases, there are multiple active treatments that we can choose from. Now, of course, there's your favorite disease that has no good treatments or only one good treatment. But if you take common diseases or even some of the rare diseases, you have multiple active treatments. So we need to be able to select from a whole buffet of options, not just this idea that there is one king and all must bow to him. And so the choice from amongst these active regimens needs to be objective. Right now, it's often based on familiarity, what is available in your country, what is affordable, those types of things. When really we could go even further and look at things like is there going to be variation in response? And that's normal, seen in almost all drugs. And so can we explain some of that variation and pick a drug with a higher probability of benefit? Toxicity is starting to become more predictable, and so we can have that as a feature. And then, of course, we have the issue that none of us go into science to really focus on, and that is that medications, especially new therapies, are very expensive. It doesn't matter whether you're in a poor country or a rich country, a major part of the expenditure is around medications. And so the idea that we can identify medications that have a best chance of controlling the disease, have a less of a chance of having systemic toxicity, and then can be done in an affordable manner is really an important element there. And so can we use precision medicine to achieve these goals is what we're really seeing a lot of growth in over the last few years, and then now as we're going forward. Now, probabilistic data is enough. In medicine, we're used to using the chance that something might happen as a way of intervening. Now, it could be something like insulin for diabetes. You don't wait until someone has gone blind from diabetes or has lost a kidney. You see uncontrolled glucose, you try exercise, you try diet. If that doesn't work, you intervene with medication of some sort. Same with statins as a primary, secondary prevention for heart disease. You don't wait until someone has had a heart attack. You intervene early, try to get the cholesterol down, try to prevent that event, decrease the probability that that event will ever occur for the individual patient. Same thing with the antihypertensives. For stroke, try to decrease the probability. You don't wait until they have a stroke. You see uncontrolled blood pressure, you try to control the blood pressure, decreasing the probability of stroke. And so that concept is really quite different than we think about for treatment. Often we're thinking about a treatment, about some sort of intervention based on the disease. But really, we're trying to decrease the chance of the disease happening at all in most instances. And I put a picture there of a young girl using the bowling lane, the 10-pin bowling, for those of you in the UK side of things. And this concept that we use the bumpers, the gutter guards, as they're also called, that can be put down. It keeps the child's ball from falling into the gutter. It doesn't guarantee a strike, but it means that they're going to hit some pins. And can we make it so that the probability of disease is dramatically decreased? The chance of going into the gutter, using this bowling metaphor, is massively decreased. And the chance of hitting some pins, having some success in terms of controlling the disease, is much higher. And so that's often what we're trying to achieve in this case. Same thing with breast cancer screening or colon cancer screening. You have age as an increasing probability of colon cancer. You reach a certain age, you start using colonoscopy or some other tools to try to identify people at risk for colon cancer. Intervene early and either have it in a benign or pre-malignant state, or even catch it early in a malignant state and decrease the chance of morbidity and mortality dramatically. And so again, the probability is decreased, therefore we can do so. And so why don't we do more of that with medicines? The adverse drug reactions are a common leading cause of death in the United States, in the UK, in the rest of Europe, in Australia, probably in every country, but not every country has looked. And so you see that adverse drug reactions are a big issue in countries like the United States and in some other countries that are heavily litigated. Many of these adverse events are now predictable. And so you always will have a situation where someone takes a dose of insulin and forgets to eat, or they take 3 pain meds because their pain is so bad and it ends up being too much for them. But often it's an adverse event that could be predicted, often by a molecular test, and therefore we can be doing better in that case. If you add new therapies, they're very expensive. And so how does one justify those? You need to enrich the probability of benefit in that case. And that equals better value, because if you're going to have to spend money, you want to make sure that for the country's sake, for the region's sake, for the individual's sake, that money is being well spent to get a gain for them in the future. And so, again, lots to do. We do have to consider what is the source of the data and the target populations on which we're building this precision medicine. And so I mentioned that because these are noble goals, are things that we can aspire to, but we need to remember the reality for this. So, for example, if you look at where drugs are developed, you think, oh, well, the drug developed with all of the world in mind. And so you're really thinking about all the different types of patients that could be benefiting from there and go forward from there. In reality, it's quite restricted. This is some figures from a paper that Dr. Todd Nepper and I had in the journal Nature back 2 years ago. And one thing that it demonstrated, I'll pop this up, is if you look at the participants of these clinical trials, these are clinical trials that were used by the US FDA as pivotal trials for the approval of a drug for central nervous system disease, for cardiac disease, and for cancer. So very common causes of morbidity and mortality in every country in the world. And so if you look at the approvals back in 1996, 92% of the participants were white, and there was some small contribution from African American or other Black individuals. Very few Asians. The whole continent was not well represented back at that point in time. You jump forward 17 years, and you see that it's 86% white, more of a contribution from Asia, not much of a change in terms of African or African American, and a few others contributing there. And so I mention this because 16% of the world's population is white. So if you're trying to take safety data from these critical, pivotal efficacy and safety trials and apply them across the world, you can automatically see some gaps. You're not going to have good representation for most of the world on these trials. And so we need to remember that as we're going forward, that the clinical trials that we're reading about in our favorite journals, Lancet, New England Journal, et cetera, are going to be very nice and well-done trials, but may not quite represent the world in the way we might need, depending where you happen to be practicing. And just to see that in a different way, here are the countries involved in the pivotal trials back in 1997. Here's what it looked like in 2004. You can see the yellow introduction, a little bit in India, more representation in South America. Here's 2009, and here's 2012. And then we have more data than that. That's the figures I put in there now. So again, not much reputation in Africa. Some increasing representation in Asia in terms of the countries that are involved, but I showed you previously the percentages are still low. So there's a ways to go in terms of making sure that the data we have can be used appropriately to decrease the probability of bad things happening, increase the probability of good things happening for patients throughout the world. Also, the way you can look at a patient to try to be more precise can range with a number of different tools. It could be tools for using nucleic acids, so DNA sequencing, DNA methylation, DNA copy number. It could be protein assays or RNA expression assays. Imaging, either a 2D imaging or dynamic imaging with functional imaging such as PET scans. Could be pharmacokinetics. Could be circulating cells. Lots of different ways we can measure a person. So I'm going to focus in on DNA because it's a practical thing. DNA is analyzed in all countries in the world. DNA is something that's quite stable and therefore can transport from a clinic in the middle of the wilderness all the way through to the big city without losing its intended desire. So it's not perfect, but it is one where we have many examples, and we'll talk about them. But remember, all these different areas and more are equally important, and someday will be more important than DNA as we make these applications. Also, when it comes to medications, certainly genes are important. You see that in the upper left-hand side here. But you have drug-drug interactions, drug-illness interactions, drug allergy interactions, drug lifestyle, drug food, many different variables that can influence whether a drug is going to work well or have an adverse event independent of the genes. And so if we really want to achieve precision medicine, we may depend on genetics as a major tool for doing so. But there are other aspects, including some very practical, easy to obtain aspects, such as asking a person about their allergies, that can be quite helpful as you move forward. Now, in terms of pharmacogenomics, the areas where the genomics is known to influence a medication, so the pharmaco for medicine, genomics for the genome, there are about 170 examples where the US FDA has included genetics somewhere in the prescribing recommendations, the so-called package insert that is available with each prescription for the medicine. But there's only a smaller group, and it's shown here, with maybe a few examples missing, that are in the dosing and administration section. So these are the sections where there is data on whether you should use the medicine, whether you should use it at a certain dose, and how you can apply it. And that includes a number of cancer drugs, showing like the top 10 or so there, and then a number of examples where it's the normal DNA and variation in that normal DNA that will influence drugs for cardiac disease, cystic fibrosis, pain control, etc., going forward. And if you take a chronically ill patient, you'll see things that really matter. Pain control, nausea and vomiting, both because of the disease and because of surgery, antidepressants, some stimulus, some blood thinners. It's not just a case where precision medicine is oncology, looking at tumor markers, but rather a lot of elements are there, including these elements that are important for cancer patients. So even if you want to be oncology-centric, you can certainly look at the tumor markers, but also how do you support the person as they're going forward? And so there's opportunities there as we push on forward. So I'm going to show you one of the examples, and it's an example of some of the data that's being generated. It's an example of some of the practical applications that are coming out, and also of the algorithms that are put together that might look fancy, but often are really quite practical as you try to apply things. So this is an example using the drug voriconazole. This is an antifungal drug. It's used in the context of prophylaxis for risk of fungal infection. It's used for treatment of some fungal infections. In the case of patients that I've been involved with, often we're using it to prevent fungal infection in patients that are having a leukemia, a myeloid leukemia in particular, where their risk of death from fungal infection can be quite high. And so introducing an antifungal like voriconazole, that medicine then can be taken, preventing a fungal infection from occurring, and has been shown to decrease mortality in that group. Well, as some of you will know, this particular drug can be broken down into inactive metabolites, or certainly less active metabolites. So you do not want this to be broken down too rapidly, or else you won't get the desired effect. And so, indeed, there are a number of genes, including those that are shown in the middle right here, including the one in red, which obviously is large and in red, so it must be the most important, that is known to influence these drugs. And so, in this particular case, there's a variant of the liver enzyme cytochrome P450-2C19, or CYP2C19 as it's called in the business. There's a variant of that called STAR17. It was the 17th variant found. And that variant is known to increase rather than decrease the amount of CYP2C19 protein that's produced, cause the enzyme to work better because there's more of it, and get rid of the drug faster. And so trying to understand if a person has the jazzed up version of this gene, the one that is going to work especially well, or a normal version of it, is important. And so ourselves and others, Dr. Kevin Hicks led this work, got an algorithm together that looked at a number of different features. But when it came down to it, you could genotype someone for the STAR17 variant of CYP2C19 ahead of given therapy, and they would either get the standard 200 milligram twice a day dose that's shown kind of in the lower middle part of the screen, or if they were a rapid metabolizer, they would get 300 milligrams twice a day, so a small increase in the dose. But if they were an ultra rapid metabolizer, they had 2 copies of the STAR17, they had super high enzyme activity, there is no dose of voriconazole you can give that will be adequate. And so they need to go to a completely different antifungal agent. And so this idea that you give 1 pill, 1.5 pills, or switch drugs is really simplistic. It's fancy genomics and fancy data, but then at the end of the day is a small change in dose or a change to a different drug that allows you to achieve precision medicine. And so that type of thing is just an example of how things are going. And when you look at whether it's been successful, if you look at those people who needed a dose adjustment, about 40% of them were within these green bars, the standard targeted range. When you adjusted the dose for genotype, those people that needed the 300 milligram twice a day dose, now after the adjustment, 80% of them were in the target range. One person was above the target range, but thankfully did not have any side effects. So it's still a good thing. And then there were 4 patients who were below the green bar for reasons that we still haven't figured out. They seem to be taking the medicines. We don't know genetically if there's something else going on, but a much smaller percentage of patients were in trouble in terms of that thing. And when you look at the overall numbers, it ends up being about 20% allele frequency in the population. So that means that about 25% of the population will have this variant. In the US white and black population, it's a little bit different numbers in Asia and in other parts of the world, but certainly in Western Europe and in the United States and Australia, you're going to see a lot of patients that can benefit from this sort of intervention because it's a very common event. 1 in 4 will need some sort of adjustment for the therapy. So that's a big number as we're going forward. You can also look at the cost, and this is data for the United States, where you looked at the budget impact analysis and identified that if you looked at the cost of a patient getting a fungal infection, it costs almost a little bit more than $30,000 in the first year extra to manage that patient because of the drugs, because of the intensive nursing, often admitted to the intensive care unit itself. A lot more expense happened with these patients. And so if you prevented a small number of these cases, you could more than pay for the entire program because of the benefits that are there. And so these sorts of economic analysis, we'll come back to them. It's really important. If you're a scientist discovering variants in genomic, in pharmacogenomic areas, if you're a person who's involved in biomarkers, you do need to understand about the economics because that's how your science can become routine much more rapidly, is by knowing how is it going to help the dollar to be stretched, the euro to be stretched, whatever it is to be stretched in a way that can benefit the population. Now, if you look at these different examples, I talked about CYP2C19, the one example. If you look at the Clinical Pharmacogenomics Implementation Consortium, or CPIC as it's shown, and also some nice data from the Personalized Medicine Coalition. But from the CPIC data, this is a group of people from about 40 different countries. There's about 300 individuals that all contribute to guidelines on whether you should, or what you should do if you have a particular genetic variant in terms of medicine. And so there are some examples, like the top right example with CFTR, where there's just one medicine that's in the guidelines. There are other examples, like the left-hand 2 down, CYP2D6 has codeine and some other pain medicines, has tricyclic antidepressants, SSRI antidepressants, has tamoxifen, has antiemetics for nausea and vomiting, has medicine for ADHD, lots of different types of medicines that are included in the same guideline. And so you can see the type of data that's emerging and growing on a monthly basis. The applications are starting to get there in terms of how does one use this in the routine care of patients and not just as part of a clinical trial. It's also important to realize that patients have multiple genomes that matter. I mentioned a previous example of patients with a fungal infection. In that case, especially if you're a patient with leukemia and a fungal infection, you have your leukemia genome, you have the fungus's genome, which can be involved with resistance, and then you also have the normal genome that is regulating a lot of the toxicities that can occur. And so in cancer, you certainly need to know the tumor genome and the germline genome, the normal tissue genome, because of efficacy and toxicity, respectively. And so this idea that we're looking at multiple genomes is really important. And so focusing initially on the idea of what do we do on the tumor side, it used to be that things were rather simple. So if you saw a tumor in the colon, was shown in the upper left-hand side, tumor in the colon was called a colon cancer. And if you looked at it under the microscope and stained it with hematoxylin and eosin, you saw some glandular formation, you called it adenocarcinoma of the colon. Or maybe in the lower right-hand side, and you sequenced it for KRAS, you now have a KRAS mutant adenocarcinoma of the colon. Simple linear type of thought. Well, that has really blown up now into a completely different paradigm. And what we're seeing now is you sequence a tumor, identify what genes are altered, and then match those to medications. And so certainly HER2 copy number in breast cancer was an early example of this. But now we're seeing HER2 copy number in lung cancer. and in gastric cancer and other cancers being therapeutically relevant. There are some genes, like the second one shown there, TP53, which does not have any drugs associated with it yet. Hopefully that'll change. And therefore is found, is of biologic relevance, but not necessarily of therapeutic relevance. And then the list goes on in terms of the medicines. But this idea that we look at what is driving the tumor and then treat accordingly is now emerging as a very viable approach, a very cost-effective approach to choosing therapy for patients. So it's not just guessing, hoping to get it half right, but seeing what is driving it and try to use it accordingly. And there's some nice data emerging around this. So this is a study that was published in JAMA a couple of years ago. What you see is an orange line that is looking at whether if you sequenced the tumor, the lung cancer, and you saw a driver, and then you targeted that driver with a therapy, you have the survival curve that's shown in orange on this plot. If you sequenced the patient's tumor and you found a driver but you did not act on it, that's shown in blue, the light blue, excuse me, and then dark blue. And then in light blue are patients who did not have any drivers, and therefore targeted therapy was not guided based on the molecular event. And what you can see is whether you have no drivers or whether you've ignored the drivers, your opportunities are worse than if you found a driver and acted on it. And it's an important part because just sequencing a tumor is not enough. Sequencing a tumor, applying precision medicine, and then making sure that you get a medicine from that is the key. And so this isn't precision genomics, it's precision medicine. You're trying to identify, can you alter care? And so you have studies like that. Here's another study that was published, a smaller study, looking at a number of different types of solid tumors. There's 12 different types of solid tumors in this particular study. And what you see here is the patients who got targeted therapy lived almost twice as long as the patients who did not get targeted therapy. One thing of note is that the economic analysis of these patients found that those who got the targeted therapy had less expense for their care, even when you included the cost of genomic testing. And so this idea that precision medicine has to be more expensive is not true, because in many cases, you're choosing the better therapy. It's controlling the disease, so keeping them out of the emergency department. It's causing fewer visits to the doctor because there's not extra visits for toxicity or such. It's allowing you to manage the patient in a way that benefits them and can be economically viable as well. So some really nice data that's being generated in that case. Now, in terms of sequencing the tissue, I've showed you some examples of that previously in that one paradigm slide. But there's also the ability to do what's so-called liquid biopsy. So when you look in the circulating plasma or serum, what you see is a bunch of different proteins, but you see a lot of nucleic acid. Every time you sneeze, some DNA is squirted into your bloodstream. And so that sort of DNA can be separated out and can be quantitated. And when you look for mutations, for alterations, you can then look and say, well, how much of this DNA is from a tumor mutation compared to what's been seen for the normal tissue, which is the majority of the DNA circulating in your bloodstream. And so this is a case where they took the tissue and lung cancer, where they took a piece of tissue, they measured it, they found two alterations, one that was actionable and where they could use this particular drug, erlotinib, one that was not actionable. A few months later, 4 months later, they did a liquid biopsy, took a blood sample, sent it out, and separated the DNA from the plasma and serum, and then we're able to measure that. And you can see some new changes that were occurring, including the alterations that is shown in yellow that is known to cause resistance to erlotinib. And so you could switch to a different epidermal growth factor receptor blocker that is able to overcome this particular genetic variant. Well, 4 months later, you look, and in gold here, or orange, you see yet another variant has emerged. This one causes resistance to osimertinib. So you now have resistance emerging the drug you switched to. You know that staying on it is not a good idea. And so in this case, there weren't other options. The ERB-B2 was not known to be actionable at that point in time. And so therefore, the patient went on classical chemotherapy. 3 months later, they were measured again. Now you have lots of different variants happening, so many that there are now enough neoantigens to justify the patient going in a clinical trial for immunotherapy, in this case, the doublet immunotherapy. And so that idea that somebody can be changing therapies based on molecular events is quite important. Now, in this particular case, this patient has now been on this clinical trial for over 2 years. You'll notice that the longest she obtained a benefit previously was 4 months on the different therapy. Now has found a therapy that is giving her benefit for multiple years. She still has PET-positive disease. It's just being kept in control. But the idea that you can use genomics to respond to emerging resistance and find new therapeutic approaches that will give the patient a better option is a key piece of what's emerging from the precision medicine era. Part of the reason it's emerging is that there are lots of different medicines that are out there. Not all of them will be available in your country, but at least in the United States, these are all approved medicines. And the point was not to make it so you can see the name of them all. The point was for you to know there's a lot of choices. If you go to a buffet and you don't really want to eat oysters, or you're tired of eating beef, there's chicken, there's fish, there's other things that you can eat at that buffet. Similar thing here. Look at what is driving the cancer, and then choose a therapy that stops that driver, is now the paradigm that we're seeing emerging, more and more data justifying its application. And so you'll be seeing that happening throughout the therapy for cancer going forward. This has caused some unique clinical issues to emerge. Some new disciplines are being developed. So for example, if you order an MRI scan or a CT scan, radiology has your back. They can help you order the proper scan. They can help you interpret the scan and go forward. You order a biopsy, the interventional radiologist can biopsy it, the pathologist can read it, they have your back. If you order sequencing, tumor sequencing in particular, who is there to help you? The testing labs do a fantastic job of making sure that there's high quality of results of what is wrong in a patient's tumor. But what do you do with it? How do you know what to do? That is where it's difficult. And so we're seeing a number of entities that have come out that are now trying to do a medical review of the pathology review. And so this is a particular company that's in the United States that does a very fine job of sequencing tumors. I have no relation there. I'm not trying to promote them. It just happens to be an example. But their reports are 26 pages long. If you want to read through all of it and try to really understand it, it can take you hours if you're not an expert in this area. So there's some helpful information on the front, but also other information that's not. In this case, the patient also got an expert molecular tumor board review. And therefore, this section in blue, this small little section in the upper middle part of there, is all that the oncologist has to read. So in less than 30 seconds, they can see what the therapeutic options are. They're ranked by medical people, and therefore they can quickly go in and apply this to their patient in a much more rapid fashion. So this idea that there are folks that are now helping translate the therapeutic review from the pathology report into the medical decision is a really important aspect that's emerging and going forward. Now, I mentioned this slide before, but I've now included the title "Take Off the Blinkers" and included this picture I found of Arnold Schwarzenegger. I have no idea why he was wearing these. I guess he was at a horse track or something. But the idea that often we're looking at one gene in isolation, maybe CYP2C19 because we're looking at voriconazole for a leukemia patient. But in reality, these genes are often measured in a panel situation. So all of these genetic variants, or all these genes, will be evaluated for genetic variants at once. And so the idea that you need to respond to having all this different, you might have ordered a test to look at one drug, but that particular test might influence a number of drugs. So CYP2C19, there's antidepressants, there's antiplatelet drugs, there's antifungal drugs, there's proton pump inhibitors for the GI tract that will all have implications for that test you ordered. But then you'll also have all the other information. So we're now seeing a need to really think about how do we look more broadly at the patient. So this little girl, this is a picture that I got off of Google Images, but as an example of a real little girl. There's a little girl I've helped care for that is a 4-year-old with cystic fibrosis. If you look forward, she is currently on or getting ready to be on anti-cystic fibrosis medicines. Cystic fibrosis patients often have some pleural pain, so they need pain control. If they need any surgical procedures or any pulmonary procedures, they're on antiemetics for the anesthesia. They often might need a blood thinner, or they might need some other antibiotics, some other medicines. Should they get depression, they need an antidepressant. You can now think about this little girl much more broadly, not only to choose which medicine she gets right away for cystic fibrosis, but to be prepared for which pain control to give her, which antidepressant should she need one, which of the antiplatelet drugs, et cetera, she's going forward. And so the idea that you can look broadly across the patient and not just at the one gene, one drug relationship is a key piece of making this stuff routine. The idea that we are focused on one gene, one drug was fine back in the day, but now we need to be thinking about what is this patient's outlook? How do we help them going forward? And that's an important part of this. Now, the last little piece, I'm going to remind us about the billing aspect. This is very American-centric, but it has relevance for the rest of the world. But if you don't understand how things are billed, you cannot use that as a stick to help the science happen faster. If you love your science and you think that it is ready to change practice, you need to then think about the economics, because it has to be practical and the economics have to work in order for things to happen. And that doesn't matter whether you're in Europe or whether you're in Asia or whether you're in South America or in North America. All of those countries, the economics drives the availability of care and the access to the medicines itself. And so it's important to look at. But we're seeing now, not only can you bill for the clinical encounter, but there are a number of other almost non-medical aspects that are emerging. So, for example, there are many different employers that are including pharmacogenomics as part of the wellness benefit, because they know that they can try to retain the patient, the employee at their company, because they're a high-tech company. They're doing wellness programs. They're doing pharmacogenomics. Therefore, they are a company on the cutting edge. You need to stay there. It also shows that you can benefit from the cost savings that occur. Lots of different opportunities. And so the employers are starting to pay for pharmacogenomics by themselves, not having the insurance company involved at all as they're going forward. And I'll skip over that just in the interest of time. You're also seeing some life insurance products where you go out there and as part of the life insurance product, should you get cancer, you now get a nurse navigator and a few other things, but you get your genomics paid for already. So you don't have to convince your doctor to submit the test and then wonder whether it'll be paid for. It's already paid for. And so this idea that life insurance is better aligned than health insurance because life insurance wants you to live longer because that's how they make their money is quite an interesting concept as we go forward. You can align it with a drug margin. So there are places where they've done the calculations. It's a particular calculator, done the calculations and identify that if they can get the medicine paid for, they will get their percentage of that as it comes back to the administration of the clinic, and therefore they'll eat the cost of the testing to get to the real money, which is in the drug reimbursement. And so that sort of thing is we're now seeing. You also see the total cost of care. So Looking more broadly, you can see panels that are looking at cross-pharmacogenomics for cancer patients, also looking at germline for choosing new therapies, and then also looking at risk of severe toxicities that can occur. And so looking broadly at this, you can start saying, well, what would the economics be? I've already mentioned about some of the pharmacogenomics, but if you take a severe toxicity like neuropathy, this is 3 different clinical cohorts: breast cancer, ovarian cancer, lymphoma. Occurs between 6 and 13% of the population. And what you see is that the patients that have, in this case, lymphoma and neuropathy, they end up costing, again, almost $30,000 extra in the first year compared to the patients with lymphoma, the same lymphoma, and no neuropathy. They also are responsible for 15 extra clinic visits compared to extra from the ones that would normally be seen for lymphoma patients. So this idea that by looking at severe toxicities, you can save some costs from severe toxicity and changing to a more expensive drug, et cetera. You can also make your clinic a little bit more open for new patients. That's causing a lot of practices now to say, all right, I'm just going to do this going forward. Now, in the interest of time, I'm going to jump over this, but what we're seeing is, how do we deliver this sort of thing? People often have multiple different doctors. People now have multiple different drugs they need to take into account. And we're seeing examples, this is just one example. I'm not trying to promote it over any others, but examples where you can have your genes, your drugs, your medical history, your allergies, your lifestyle, be able to put into medicines regardless of which doctor prescribes them, and be able to manage them in a more holistic fashion. And so we're seeing tools like this emerge and starting to be applied going forward. So I'll end up going and closing with this slide that just is a reminder that lots of different examples are there in terms of precision medicine, and even more are coming. Your favorite disease or your favorite drug is next in terms of trying to apply that. And so we have, again, examples where we can now use select therapy among equals. The idea that there's a best therapy and second best therapy is really outdated. Most of the time, we know there's a bunch of really good therapies. We don't know that one is better than another for an individual person unless we look and try to identify risk of toxicity, risk of other things. Areas like pediatrics, they never have level 1, type 1 evidence-based medicine. They always have to go with small studies or make inference from adult studies. And so many areas of medicine do this. And so the idea that we can use this and apply it is critical. Rational therapeutics, risk mitigation, the budget impact, all can be influenced by precision medicine, and are areas where the accountants and the administrators, who are often the obstacle to new innovations, can be influenced, because they now are going to realize, hey, we can do better. We can be a better hospital. We can save costs. We can keep our doctors, keep our nurses, keep our pharmacists. That sort of thing is influenced by this. Now that we're looking across the patients, we do something for them today, maybe in a hospital. How are we going to make sure that they benefit from this information when they go to their endocrinologist or their primary care physician or whoever it is they might see in the future? Again, different ways of discharging patients, thinking about their needs, different ways of doing discovery in terms of what's the best way. And then lastly, we need to make sure that we're asking the patient about the toxicity. If I think that toxicity is severe, it doesn't matter. It's what the patient thinks. I might think it's fine, the patient thinks it's terrible, or the patient that we're trying to help, not the clinician, not the researcher, not that. So we need to be asking them. And with patient-derived outcome data, this can feedback, can allow us to really direct the care going forward to really achieve things in a much better way. So this is the last slide. I'm going to stop at this point. I hope you've picked up a few little tips and nuances as we went along. There's a great rest of the session that's coming up right after this. And it's really an exciting time to look at this week, and really the broad spectrum of topics that LabRoots is putting together, and trying to make it so that we can enhance our knowledge and make our research better, make our clinical care better, make things better for those that we care about. So I want to thank you very much for your time. Thank you.